Okay, Assalamualaikum dan salam sejahtera. Saya ucapkan kepada Yang Berusaha Profesor Madya Dr. Norrizah Jaafar Sidik, Yang Berusaha Dr. Khanizah Hasliza Abdul Khalil dan Barisan Pensyarah Biologi. Saya Puan Marisa Muhammad akan menjadi moderator anda untuk sesi syarahan biologi yang akan dijalankan secara langsung di YouTube Pusat Asasi. Sebelum saya memperkenalkan siapakah pensyarah jemputan pada kali ini, kita mulakan dengan bacaan doa. Dengan ini saya menjemput saudara Wan Muhammad Hadif bin Wan Syukri dari kumpulan S6. Silakan Hadif.
Bismillahirrahmanirrahim. Al-Fatihah. A'udzu billahi minasy syaithanir rajim. Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin. Arrahmanir rahim. Maliki yaumiddin. Iyyaka na'budu wa iyyaka nasta'in. Ihdinas siratal mustaqim. Siratal ladzina an'amta 'alaihim ghairil maghdubi 'alaihim waladdallin. Bismillahirrahmanirrahim. Alhamdulillahi Rabbil Alamin. Wassalatu wassalamu ala ashrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Allahumma aftah alayna hikmataka wanshur alayna min khazaini rahmatika ya arhamar rahimin. Rabbana zidna ilma warzuqna fahma waj'alna minas salihin. Wa sallallahu ala sayyidina Muhammad وعلى آله وصحبه وسلم والحمد لله رب العالمين.
Terima kasih saudara Hadif. Untuk pengetahuan para pelajar, sesi program ini adalah untuk memberi peluang kepada anda untuk mendapat pengalaman menghadiri syarahan yang dijalankan oleh pensyarah-pensyarah dari fakulti. Semoga pendedahan ini akan dapat dimanfaatkan oleh semua. Dengan ini saya memperkenalkan Profesor Madya Dr. Norrizah Jaafar Sidik. Beliau sekarang bertugas di Pusat Pengajian Biologi, Fakulti Sains Gunaan UiTM Shah Alam. Beliau adalah pemegang ijazah dan sarjana dari Universiti Malaya dan dianugerahkan Dr. Falsafah dalam bidang Plant Teknologi dari Universiti Putra Malaysia. Beliau mempunyai pengalaman mengajar dalam bidang Research Methodology, Plant Physiology, Biosafety dan Biosecurity, Contemporary Biology, Biodiversity, Botani dan Animal Physiology. Selain itu, beliau turut menyelia pelajar Paskah, Siswazah Tempatan dan Antarabangsa. Beliau adalah pakar dalam bidang Plant Biotechnology. Profesor Madya Dr. Norrizah Jaafar Sidik adalah salah seorang daripada ahli dalam Excellent Entitled Agriculture Biotechnology Research Group UiTM, International Society for Horticulture Science, Natural Products Society dan Malaysia Society of Plant Physiology. Dengan ini, saya berjemput Profesor Dr. Norrizah Jaafar Sidik untuk menyampaikan syarahan beliau untuk bab 3. Silakan Dr. Norrizah.
Okay, Assalamualaikum semua. Okay, saya ucapkan terima kasih kepada Puan Marisa. Okay, so hari ini saya akan memberi syarahan berkaitan dengan plasma membrane. Okay. Jadi saya akan share presentation slide sekarang. Okay, so today's topic, I will give lectures on the plasma membrane transport, right? So for this topic, there are 3 parts, part 1, part 2 and part 3. Okay, so after every part, there is a break, yeah? And I will go for the first part first, okay? So before that, I will show you the learning outcomes, yeah? So at the end of the course, students should be able to list the components of plasma membrane and their functions, describe the structure of the plasma membrane and the functions of each of its components, explain the role of membrane protein, explain how membrane structure results in selective permeability, explain the various transport mechanisms across the membrane. These are the learning outcomes. Then we go to the membrane components and its organization. So if you look at the figures here, on the right side, okay, it shows the plasma membrane structure where it composed of a bilayer of the phospholipid, okay, interspersed with the proteins, right? So this plasma membrane can be found in the prokaryotic cells and also in the eukaryotic cells. So if you look at the figure here, the plasma membrane is just beside the cell wall, okay? So prokaryotic cells, example like the bacteria, and also it also can be found in the eukaryotic cells, okay? So unlike prokaryotes, the eukaryotic cells also possess internal membranes that encase their organelles and control the exchange of essential cell components. Okay, now we look at the functions, the plasma membranes. One is to separate living cells from its surrounding. Okay, the plasma membrane, it provides protection for a cell, okay, and act as a barrier. It also provides a fixed environment inside the cells. Okay, so plasma membrane also, it controls traffic into and out of the cell it surrounds. Okay, so plasma membrane can transport ions, nutrients in and out. Okay, plasma membrane regulates not only what enter the cells, but how much of any given substance comes in. Okay, and it also exhibits selective permeability. Selective permeability, it refers to its ability to differentiate between different types of molecule, only allowing some molecules through while blocking other molecules or substance. Okay, so it only allow certain substance across. Okay, and the plasma membrane, okay, it contains the phospholipid. The most abundant lipid in the plasma membrane are the phospholipid. Phospholipid are amphipathic molecules. Amphipathic molecule meaning that it contains the hydrophobic regions and also the hydrophilic regions. Now we look at the pictures here, right? So these are the phospholipids, yeah? So the phospholipid, it has the hydrophilic heads, where the hydrophilic heads is a membrane bilayer where it faces outward, contacting the aqueous or watery fluid both inside and outside the cells. Yeah? Since water is a polar molecule, it readily forms electrostatic charge based interaction with the phospholipid heads. And then have the hydrophobic tails. Yeah? These hydrophobic tails are water-fearing. Okay? It consists of a long non-polar fatty acid tails. The fatty acid tails can easily interact with other non-polar molecules, but they interact poorly with water. Okay, so this is about the phospholipids. Now we look at the structure of the plasma membrane. Okay, it shows here in the diagram the fluid mosaic model. Fluid mosaic model, where this model is proposed by S. Jonathan Singh and Garth Nicholson, in 1972. According to the fluid mosaic model, the plasma membrane is a mosaic of components. You can see here, mosaic components. Primarily is the phospholipid, and then we have the cholesterol, okay, Okay, if you look at this one, okay, you can see here the cholesterol, okay, cholesterol, and then it has the proteins, the proteins here. Go back to the figures here, the proteins, all these are the proteins that move freely and fluidly in the plane of the membrane. Okay, so it is a dynamic process in which phospholipids and protein are continually sliding past one another, right? So now we look at the components in detail. Okay, we look at the phospholipid here, the main fabric of the membrane, and then we have the cholesterol tucked between the hydrophobic tails of the membrane phospholipid. And then we have the integral protein. Okay, the integral protein is embedded in the phospholipid bilayer, may or may not extend through both layers. And we have another one is the peripheral proteins. Can see here on the inner or outer surface of the phospholipid bilayer, but it's not embedded in its hydrophobic core. And then we have carbohydrates here. These attached to proteins or lipids. Okay, if it is attached to the proteins here, then call it as glycoprotein. When it is attached to the lipids here, then we call as the glycolipid. Okay, so these membranes, they are not static. They move freely, dynamic, which are not firmly fixed in a place. They are flexible and fluid motion. Okay, so membranes are not statics. So they are, yes.
Can I ask question? The term glycoprotein, glycolipid, is it refer to the combinations of protein for the glycoprotein? Does it refer to the combinations of the protein part?
Yes, this one protein, right? Protein with the carbohydrates, right?
Okay, yeah.
Glycoprotein, right? Whereas here, the phospholipid with the carbohydrate. Then we get the glycolipid.
Okay, thank you.
Okay, welcome. And then we have, okay, so we look at the structure of the plasma membrane. Okay, it's fluids. It's membrane are not static. Yeah, they are not static. They move freely, right? And flexible, fluid motion, right? So, what are the factors that affecting the fluidity? Okay, it's stated here. Okay, the one is the movement of phospholipid molecules. The second one is the presence of unsaturated fatty acids, and the third one is the presence of the cholesterol, right? So before we move to the next slide, I just want to explain the phospholipid. Yeah, so the head contain of the phosphate and the glycerol. Yeah, so these are the hydrophilic head, and this one, the tail, it has two tails. Yeah, so it is the saturated fatty acid. This is the straight tail, right? And saturated fatty acid, they have no double bonds. Okay, no double bonds. They are saturated with hydrogen, so they are relatively straight. But the next one, the second tail, these are the unsaturated fatty acid. Unsaturated fatty acid, it contains one or more double bonds often resulting in a band. So you can see here, there's a band here or kink, what we call as kink, right? Okay, now we look at the next slide, movement of the phospholipid molecules. Okay, it held together by the hydrophobic interactions. So phospholipid molecule carry out flip-flop movement across the two layers of the phospholipid. So now we look at the structure of the phospholipid here. This shows the flip-flop. The flip-flop phospholipid flip-flop. It moved to the other side layer. It can flip to the flop to the other side. So this occur once per month. Once per month. And then we have another movement, what we call as lateral movement. Okay, this lateral movement phospholipid is within the same layer. Yes, so you can see the arrow here. It occurs within the layers. Yeah, and this lateral movement occurs about 7 to the power of it occurs 10 to the power of 7 times per second. And there is another movement. Okay, the proteins. The protein move laterally, right? Okay, so it is next we go to the temperature, which affects the phospholipid movement. Okay, and how close together they are found. When it's cool, okay, they are found close together, and when it's hot, they move further apart. So this is without cholesterol. Okay, cold, rigid. It's not as fluid, may break. Okay, but during hot temperature, it's too fluid, flexible, won't hold the shape. But this is without cholesterol. Okay, and then another factor that affecting fluidity is the presence of unsaturated fatty acid. Okay, so as I mentioned just now, unsaturated fatty acids. Okay, look at the tail here. The phospholipid tail. This is the unsaturated fatty acid. Okay, so as temperature cools, membrane switch from a fluid state to a solid state. Solid state. Okay, this is when the temperature cools. So the temperature at which a membrane solidifies depends also on the types of lipid it is made. Okay, now we look at the presence of still discuss on the presence of unsaturated fatty acids. The membrane rich in phospholipid with unsaturated hydrocarbon tails. Okay, it remains the fluid at lower temperature. Okay, so unsaturated hydrocarbon tails cannot pack together as closely as saturated hydrocarbon tails when we compare between these two, because due to the presence of double bonds that form the kinks in the tail. Okay, so the membrane are more fluid. You can see here, right? The kinks here, the kinks, unsaturated hydrocarbon tail with the kinks. So unsaturated hydrocarbon tails of phospholipid have kinks that keep the molecule from packing together, right? So the membrane will be more fluid, like the salad oil, become fluid. All right. Now we look at the this picture. This is the winter wheat. Winter example here where the lipid composition of the cell membranes can change as an adjustment to changing temperature. Right? For example, given here is the winter wheat. Yeah? How this plant tolerate the extreme condition temperature is by increasing the percentage of the unsaturated phospholipid in autumn. Okay, so this keeps the membrane from solidifying during the winter, during the cool temperature. Okay. Okay, another example given here is the fish. Yeah, they are capable of adapting to cool environments by changing the proportion of unsaturated fatty acid in their membranes in response to the lowering of the temperature. Okay, another factor that affecting fluidity, yeah, is the cholesterol, right? Okay, in animals, yeah, the cholesterol keeps the membrane fluid. Okay, so it lies. Okay, the cholesterol, it lies alongside the phospholipid in the membrane and tends to dampen the effects of temperature on the membrane. Okay. Okay, right. So the cholesterol has different effects on membrane fluidity at different temperature. Okay, so you can see here, the cholesterol is between the phospholipid, wedged between the phospholipid in the plasma membrane, okay, of the animal cell. So during high temperature or warm temperature at body temperature 37 degrees Celsius, okay, the cholesterol will restrain the movement of phospholipid and the membrane will become less fluid. This is during the warm temperature. Whereas during the cool temperature, it maintains fluidity by preventing tight packing and slow down the solidification of the membrane, right? Okay, so you can see here the cholesterol, the presence of cholesterol within the phospholipid here, in the animal cell membrane. So cholesterol also, it reduces the membrane fluidity at moderate temperature, okay, by reducing the phospholipid movement. At low temperature, it hinders solidification by disrupting. Okay, by disrupting it, it will interrupt. It hinders solidification. It inhibit solidifications, or it obstruct solidification by interrupt the regular packing of the phospholipids. Okay, so the cholesterol also acts as a fluidity buffer for the membrane. It resists changes in membrane fluidity caused by changes in temperature. Okay, so it functions. Cholesterol functions as a buffer, preventing lower temperatures from inhibiting fluidity and preventing higher temperature from increasing fluidity too much. Okay, so cholesterol extends in both directions. Okay, the range of temperature in which the membrane is appropriately fluid and consequently it function. Okay, functional. Okay, so membrane must be fluid, fluid to work properly. Okay, if it is not fluid, then the membrane cannot functions, cannot work properly. Okay, so when a membrane solidifies, its permeability will change. Okay, the enzyme protein in the membrane will become inactive. That's why membrane must be fluid. Okay. All right. So this is the last slide for part 1. Okay. There are some questions. Okay. The first one is list 3 components of plasma membrane. Okay. And then the second question is name 2 types of phospholipid movement. The third one is why is the plasma membrane described as fluid mosaic? Okay. The last question is how unsaturated hydrocarbon chain maintain the membrane fluidity. So students can answer the questions. You can refer to the these slides, or you can also get more information from the textbook. Okay, so this is the end of my lecture in part 1. Thank you.